you. Thank you, Jerry, for that kind introduction, and thanks to all of you for coming today. I am, as Dr. McNeil just mentioned, an evolutionary geneticist. I'm an assistant professor in the biology department in my third year. And as an evolutionary geneticist, I'm fascinated by the ways in which the biodiversity that we see around us on our planet has been created. And so today what I'd like to do is to talk to you a little bit about where that biodiversity comes from, and in particular using a case study of fishes in the lower Congo River of Africa as a model for how we can study the processes that create diversity in our world. I like to begin all my biology classes with this quote, this wonderful quote from Theodosius Dobzhansky: "Nothing in biology makes sense except in the light of evolution." So I am putting this quote up here today both as a shameless plug for my evolution course that I'll be teaching in the spring of 2013. So for any bio majors in the crowd, please consider enrolling. It'll be a wonderful, wonderful course. But also just to remind us that evolution really is the backcloth for all of biology as we know it. So in many of the biology courses that we take, we study the way in which things work, sort of the mechanisms, whether at the molecular or the organismal or the ecological level. But the philosophers in us and the poets in us want to know where all these neat systems came from. If you look at the eye, we want to know not only how it works, but how it evolved. And so that's one reason why I really love this quote from Dobzhansky, who was a famous geneticist who really brought together some of Darwin's ideas with the then emerging field of genetics. So today I'm going to start out talking with you about what species are, how they originate, so some of the theory that's emerged about how species are formed. I'll talk a little bit then about a system that is new to me. So I've been working on this project for about 2 years now, and it's just been an incredibly exciting project to work on, Lower Congo River fishes. And then I'll talk a little bit about some of our new data on genetic evidence for the mode of speciation in the Lower Congo River. I want to make sure to mention that all of the work that I'll be talking about today is in a deep and close collaboration with two folks, Melanie Stiasny, who's an ichthyologist at the American Museum of Natural History, and who's really the leader of the team of scientists that are working on issues in the Lower Congo River and its biodiversity. And then Jeff Markert, who's an ichthyologist at Providence College, who's also a close collaborator. Just to give you a taste of what's to come before we delve into some of the more technical aspects of species and speciation, this slide will give you an idea of some of the incredible morphological adaptations that are present in the Lower Congo River, some of the sort of bizarre morphologies that we see in different fishes. The Lower Congo River is a place of rapids and waterfalls and eddies, just incredibly intense hydrology, and I'll tell you a lot more about that as we move along. But this gives you an idea of some of these strange fish species that have been discovered in this system. And it also gives me the opportunity to mention that many of the images that I'll be showing come from this wonderful website from the American Museum of Natural History. And I urge you to take a look at it if you have a spare moment. The Congo River Project has a really extraordinary collection of photos and videos documenting this unique ecosystem. All right, so let's start out with a discussion about what species are. Many of my students are surprised to learn that biologists don't agree about the definition of a species. There are, I've heard it said there are as many definitions of a species as there are evolutionary biologists out there. Everyone seems to have their own kind of pet definition, which may lead you to the understanding that species, the term species, the concept of a species, is not necessarily a fixed biological entity, but is rather, we can think of as sort of a human construct. So because of that, we do get this quite a wide variety of definitions of what a species is. It may be sort of like art. The concept of art, we don't necessarily all agree on it, but we all think we know one when we see one. Okay, so let's go through a couple of these definitions. The most basic one that we can probably dispense with pretty quickly is what's known as typological or phenotypic species. So these are species that or a species concept that says that things that look alike all belong to the same species. So it's very convenient, of course, and easy, and it's the oldest definition of species. However, we can, like I said, dispense with it pretty quickly. We can think of examples from nature that violate this notion of a species concept very quickly. For instance, take the eastern and western meadowlark, which look nearly identical, so you wouldn't be able to distinguish them even as a naturalist. They have overlapping ranges, yet they have different songs, and those different songs prevent them from breeding with each other, from interbreeding. We can take also the case that's probably even better known to you of the wide morphological diversity of dogs. So if someone asked you if a Chihuahua and a Great Dane belong to the same species, you'd say yes, even though if you were an alien from another planet and you looked at these things, you'd say they look completely different. There's no way I would group those together. So we're getting towards this concept of reproductive isolation, the inability to interbreed, as an idea about how we should define species. And sure enough, the next big species concept to arise, the biological species concept, is focused around this idea of reproductive isolation. So Ernst Mayr, who was a famous evolutionary biologist who died relatively recently, coined the biological species concept, which focuses on the inability of species to reproduce, either in actuality or potentially. So either actual or potential inability to interbreed causes isolation, which leads to true biological species. However, once again, we can think of examples in nature, quite a few, that violate this species concept. So the biological species concept, while it's, you can think of it as kind of like it's sort of like the layman's version of what a species is. Most biologists do not feel comfortable with this species concept because of examples like this. So take a species of coral that lives in the Caribbean, very common species of coral in the Caribbean, called Acropora prolifera. So genetic studies have shown that Acropora prolifera is actually a hybrid of two other coral species. Okay? It's not a hybrid that can itself reproduce sexually, so in that sense it's like a mule. However, because corals reproduce themselves asexually as well as sexually, they can reproduce clonally and continue growing sort of in perpetuity. This particular morphologically distinct kind of coral is genetically different from either of its parents and can continue existing potentially eternally. So this is why it's been termed the eternal mule. And we've recognized this Acropora prolifera as a species for many decades, but under Mayer's concept of biological species concept, it would not count as a species. We can also think of another common biological system that violates the notion of biological species concept, and this is a ring species. So a ring species is a group of organisms that are found in a geographic ring, and here we have Insatina salamanders in California, and each of these subspecies of salamander is slightly genetically and slightly morphologically distinct from the one that is next to it. However, there is interbreeding between all the adjacent subspecies until you get all the way around the ring and you get two subspecies, it's here, Clauberry and Eschscholtzi, that do not interbreed. So these we can say, okay, these are good species under the biological species concept, but what about everything in the middle? Where do we draw that line? So you can start to see how again and again nature is resisting our attempts to shoehorn it into these species concepts, these classifications. So that brings us to the third sort of larger category of species concepts, which we can group together as lineage species concepts. And most modern species concepts that biologists feel good about are, in one form or another, lineage concepts. So what all these different concepts have in common is that the notion of a species is one of a lineage that is evolving independently from other lineages. Where disagreement may come in is what constitutes independent evolution. How do we measure that, and how do we prove that one lineage is truly evolving separately than the others? One thing to note here is that lineage species concepts, while they may require substantial reproductive isolation, they do not require total reproductive isolation. All right, so now that we've talked a bit about various species concepts, what a species is, how do those species come about in the first place? How do they form? Well, we can think of this in terms of the sort of the rubric we just set up. What are the processes that could cause substantial reproductive isolation between populations, that is, between nascent or incipient species? So we'll go through a couple of the biggest hits of these forces that are thought to be responsible for causing reproductive isolation. The first of which is genetic drift. Okay, so genetic drift we can think of as random change in a population over time. So genetic drift does not imply that any kind of selection is going on, any kind of natural selection or sexual selection is going on. It's also influenced both by population size and by gene flow. So smaller populations are much more influenced by genetic drift than larger populations. If you have two populations, say of beetles, where they are genetically different from each other, if you have individuals that are migrating between one population and the other, we call that gene flow. Okay, so if one individual is migrating from one population to the other and is breeding with the individuals in this population, we term that gene flow. Gene flow is a homogenizing force that will prevent those two populations from diverging from each other. Okay? So genetic drift is our first big force that's causing reproductive isolation, but it doesn't invoke any kind of selection. That is, the environment is, you can think of as sort of irrelevant. All right, so that brings us to natural selection, the next big force. Natural selection is probably more what you had in mind when you started thinking about what causes two populations to diverge from one another. So natural selection, of course, is the process by which some individuals in a population, because of a particular difference that they have from other individuals in the population, are better able to reproduce. And that difference may be, for instance, that a mutation caused a difference in morphology in their jaw, such that they were able to feed by sucking instead of biting, and thus exploit a new food resource in the lake where they live, like sucking algae off a rock. So they were able to get more food, they were able to have more babies. Those babies inherited their same jaw morphology, et cetera. So natural selection thus encourages isolation, reproductive isolation, and the process of speciation. And then the third major force, which we won't talk much about today, but is kind of finishes up the trio of the important forces causing speciation, is sexual selection. So imagine a mutation in an individual of a population of fishes that causes a particular female fish to be more attracted to males that have very big tails. Okay? So you can imagine how that sort of mutation would cause kind of a reinforcement to take place over time that would rapidly cause divergence between those fishes that had that mutation and other fishes that did not. Okay, so that brings us then to the ecological or biogeographic context of speciation, and this will turn out to be very important for the study system that I'm going to describe in just a moment. So how does the environment influence speciation? Well, as you can probably tell, evolutionary biologists love to classify things. So speciation events have been classified depending on the sort of the biogeography in which the events are occurring. So the easiest example that we can think of in this biogeographic scheme is allopatric speciation. So allopatric speciation occurs when two populations are separated by a geographic barrier. So we can think of our population of beetles again. We can think of a stream that all of a sudden is diverted and starts running right through the middle of this population. Now the beetles can't cross the stream, and so they start to separate by genetic drift. There may be an element of selection in there, but please note that there does not have to be an element of selection in there. So these two populations can diverge purely by genetic drift, just by random chance. And when they come back together, for instance, if the river dries up, they may have evolved to an extent that they're no longer able to breed with one another. There's a great example of allopatric speciation that I thought I'd mention. This is an example from Nancy Knowlton's team at Smithsonian Tropical Research Institute. They study a kind of shrimp called Alpheus shrimp. And they were very interested in knowing whether species of shrimp that were found on either side of the Isthmus of Panama, so on either side of the Panama Canal, on the Pacific side and the Atlantic side, were in fact sister species. So were all these species created by the rising of the isthmus about 3 million years ago? And when they looked at the genetic signature of those species on either side, they found something very interesting. So here you can see a phylogeny. This is a species tree. All the names in red are from the Atlantic, all the names in blue are from the Pacific. So for each of these clades, you have essentially sister species on each side of the Isthmus of Panama. So that is just a beautiful example for allopatric speciation. There were all these different populations that were essentially separated by the rising of the Isthmus of Panama and diverged over time to become different species. But we can see very nicely their genetic relationships in this phylogeny. All right, so that's sort of the easiest example. Now, coming together sort of geographically, more closely in space, the next mode of speciation is called parapatric speciation, and this is when you have speciation that's occurring while there's still gene flow occurring. So the populations are still proximate in space; they're not totally geographically separated from each other. Gene flow, some amount of gene flow can still occur. So we can think of, for instance, the Ensatina salamanders in California that I mentioned, where you do have gene flow occurring at each step. This diagram here shows something very similar. So we can imagine a population of beetles where they're spread out across a range. Beetles that are closer in space to each other, that are sort of at one end of the range, will have a much higher percentage or probability of breeding with each other than beetles that are found at totally opposite ends. And over time, that could lead to enough divergence between the ends of that range that you get different species. And then finally, the last mode of speciation is also sort of the most contentious and the most mysterious, and this is sympatric speciation, species that arise in sympatry. In other words, in the same place. So no geographic separation. You can think of it as reproductive isolation without geographic isolation. And there are large volumes of theory trying to work out exactly how this could occur. We have a few examples of it in nature, including lake cichlids, which I'll mention in just a minute. You can think of, for instance, the sexual selection example we were talking about. Fish could be in the same lake, but a mutation could arise that causes them to segregate themselves out in some way. All right. So having then reviewed all these different modes of speciation, what we're really ultimately interested in is what is the genetic basis for that speciation. So how quickly is divergence occurring? We can measure that using genes. Along which geographic ecological axes? We can measure that by looking at different populations. How many genes are involved in speciation? In other words, if you have some extent of mixing, are there particular genes that sort of pull more weight than others in the speciation process? And finally, how important is genetic drift, so relative to selection in generating species? So if you have some degree of gene flow, some degree of migration between populations, how much migration do you need to have before it swamps out natural selection that's pulling those species apart? So we can measure genetic drift in incipient species with genetic data. This sort of brief diagram just shows that you can go from genetic data, you can build genealogies, you can use those genealogies to assess various aspects of demography that tell you something about genetic drift. So long-term population size, effective population size, population dynamics over time, and gene flow or migration. These are all things that influence genetic drift. So we can get an idea of those characteristics using genetic data. And likewise, we can look for, this is much trickier, but we can also look for signatures of natural selection in the genome using genetic data, as well as morphological data, which is the more traditional way to do it. So an example here is two color morphs in white sands lizards the color in their skin has been shown to be controlled by a single gene, the MC1R locus. And this is sort of a famous example of where this one gene has permitted lizards, a population of lizards that lives on a white sand background, to quickly adapt to that environment. And of course, the selective pressure there is predation. A white lizard blends in much better on a nice white sand background than the dark lizard. All right, so that brings us to the study system. Here, we're interested in understanding what factors promote speciation in freshwater fish, particularly in the tropics. Now, if you've taken any evolutionary biology or probably general biology, you've heard a bit about cichlids. So cichlids are famous because they are the most speciose family of vertebrates, period. Okay, that's saying something. They're extraordinarily speciose, more species of cichlids than any other family of vertebrate. And they've been extraordinarily well studied, so many of the models of things like sympatric speciation focus on cichlids in the Great Lakes of Africa. And they are deservedly famous because there's very interesting processes that have been documented there that include color morphs, sexual selection, selection on particular jaw morphologies. However, other systems in the tropics, notably rivers, have received a lot less attention. And this is partly because rivers tend to be much less diverse than lakes in general in terms of the freshwater fish diversity. But one exception is the Lower Congo River. So the Lower Congo River is an amazing place. It is very unusual among freshwater rivers, even in the tropics, for its high degree of diversity and its high degree of endemism. So endemism, you may recall, is essentially a measure of species that live in that place and no other place on Earth. So in the case of the Lower Congo River, there's over 320 species that have been described from, or 320 species found in this river, and over 90 of them are found no place else. So it's a remarkably high rate of endemism. I mentioned that the Lower Congo River has this extraordinary hydrology. So my colleague, Dr. Stiasny, likes to refer to it as a place of superlatives, and it really is. It drains an area about the size of Europe and forces all of that water through a series of narrow channels, waterfalls, eddies, just produces these incredible rapids. The Lower Congo proper begins in a sort of a lake-like area, which is now called Pool Malebo. It used to be called Stanley Pool. So this is near the twin cities of Kinshasa and Brazzaville. And from there, it falls over a sill and drops about 280 meters over about 350 kilometers as it makes its way to the Atlantic. So you can see some of these great elevational drops that occur along its route. And it's our belief that it's this extreme hydrology, these rapids, these eddies, that are acting as essentially features promoting speciation in this environment. So what we're really interested in is the mode and rate of speciation in the lower Congo Basin. What is it that's creating this extraordinary diversity and endemism? So the ultimate question here, which seems like kind of a funny question on its face, is can water act as a barrier to fish? Could it be water that's causing allopatric speciation in these fish? Is the water in the Congo essentially like a mountain range for populations on either side, causing divergence and diversification? This picture gives you just a little bit of an idea of the scale of some of these rapids. This is a kayaker here. Imagine yourself as a little fish trying to cross that river.
Is that you?
No, some of the collections that have been done, though, have brought in professional kayakers by necessity. So this is Dr. Stiasny and some of her team in the Congo collecting fishes. As you just saw, the conditions make it, both the hydrological but also the political conditions can make it quite a challenge to get access to the samples there. But they've been collecting for about 4 years, and then I've been on board for the last 2 years doing the genetic work, the boring work in the lab. I wanted to mention, as long as we're talking about the hydrology, that these questions are not purely academic in nature. So there is a massive hydroelectric project that's proposed for the lower Congo River. It's called the Grand Inga Hydroelectric Project, and as it's proposed, it would be 2 times the size of the Three Gorges Dam in China, which is currently the largest dam. It's proposed for a waterfall called Inga Falls, that's by volume the largest waterfall in the world. And this dam would divert probably about 2/3 of the water. So it's a very interesting question, both in terms of what it would do to the ecology there, but then balance that with the electricity that would potentially provide in a very power poor region. But I wanted to mention that as sort of a real-life application of these biodiversity studies. All right, so our studies right now are focusing on a narrower range of the lower Congo River. So we have samples and study sites throughout the whole lower Congo River, but right now I'm going to tell you about samples that have just been collected from this region. So this is Pool Malebo, also known as Stanley Pool, and you may remember there was a big drop that occurred right after Pool Malebo. So these are the Kinsuka-le-Rapide Rapids. It's the first true rapids of the lower Congo River. So we are collecting species that have populations on both sides of this river and looking to see whether we see any evidence that those species or those populations are diverging from one another. In other words, are we looking at incipient species? So that would give us evidence that in fact water is acting as a barrier to these fish. We also suspect that there are some features, some inherent features of fishes that may make them more or less prone to speciate. And so we're taking kind of a comparative approach for that reason. So we're looking at morphology, the body shape of the fishes. Do they have rheophilic features? So rheophilic features means adapted to rapids. So some of the rheophilic features you can think of are compressed body shape, like very flat, anything that would make it easier to stick to rocks. Very big pectoral fins. So the fins on the side here are the pectoral fins of the fish. Very big pectoral fins make it easier to swim in rapidly flowing water or to stay in place. And then reduced swim bladder. If you don't need to be going up and down in the water column, then you're going to reduce your swim bladder. Swim bladder is a fairly expensive physiological organ. So all of those may limit movement. Relative size of the fish, if you're a bigger fish, you're going to be able to cross rapids more easily. Habitat specialization could also reduce movement, as could being a mouth brooder versus fish that spawn on substrate. All right, so our overall objective here then is to investigate the role of these hydrological barriers in generating fish biodiversity. That's sort of the first stage of this project, and that's what the data that I'll show in a moment. The next stage is going to be to move towards some new sequencing techniques in order to get a very fine-scale genetic map of these different species to look for the genes that are involved in speciation. So that will allow us to gain this extra access of being able to look for clues about natural selection and how it's acting on these fish. This is just a list of some of the cichlid species we've collected in the Lower Congo River. And here's the ones that I'll show you some data for today. Okay, so what I want to note here is that we have a diversity of species that live in, some of which are habitat specialists, some of which are more generalist, some are larger, some are smaller, and they have different modes of reproductive behavior. So again, the ultimate goal is this comparative approach to look at the patterns we see across all these species. The first species that I'll mention is Orthochromys stormsii. So this is a medium-sized fish that is highly endemic to the region even within the Lower Congo. It's found in a very narrow band, only in the Kinsuka-Les Rapides area. And it has a reduced swim bladder, but you can see it's not compressed the way some of the other fishes that I'll show will be. It has pretty strict habitat requirements. It likes still waters and backwaters that are removed from the main channel. So when we look at the genetic types that we find in this fish, and I won't go into any great detail about the data set, but I'll just say that we have about 2,000 base pairs of mitochondrial DNA. So what you're looking at here is a haplotype network where each circle represents a different genetic type. And the color represents which site it was collected from. Okay? In red, those fish were collected from site A, Kinsuka, and blue is site B, Les Rapides. Okay? And I should mention too, these sites are separated by only about 1.5 kilometers. So very, very short distance as the crow flies. And despite that, we see this enormous difference between Kinsuka and Les Rapides. So this network shows you how closely related each genetic type is. In other words, this genetic type is only one genetic mutation away from this one, whereas this one's quite a bit further away, telling us that they've been separated for longer. Okay? So the fact that we see all the red in one area and all the blue in another area tells us that these populations, in fact, are on different evolutionary trajectories. So it supports this notion that hydrology is essentially acting as a barrier to these fish. Let's look at the next species. So this is Teleogramma richardii. Of all the cichlid species found in the Congo, this one is the most rheophilic. We see essentially all the classic hallmarks of a rapids adapted fish. Very compressed body, very compressed cranium, huge pectoral fins here, very reduced swim bladder. It's got a bit of a bigger range than the Orthochromys, but nevertheless, we see this extraordinary differentiation between sites. So this is even stronger than what I just showed you. We see all the genetic types from site A clustering in one area, all of them from type B clustering in another. Okay, so again, further evidence that in yet another species we have this separation. This I won't talk about too much, but this is data from microsatellite markers that essentially shows the same thing. So these are additional parts of the genome that are telling us the same story. Dr. Stiasny, our colleague, has done some morphometric work on these fish as well. And in accordance with their genetic separation, she has also determined that there are some differences between, for instance, the caudal fin morphology in these two populations. So not only are they genetically different, but there are some morphological differences that we can see as well. And then the third fish that I'll mention is Lamprologus werneri. So this is a substrate spawning cichlid. Again, you can see it's got a pretty flat body shape, reduced swim bladder again. And once again, we see that we get this pretty strong differentiation between sites. However, it's not as strong as in the other two. But we do see that the difference is confirmed by microsatellite markers, so by other parts of the genome. The genetic difference is not as strong, so we might not expect there to be as much morphological difference. And in fact, when Dr. Stiassny has looked, she has seen no evidence for morphological, morphometric difference between these species. And then finally, I wanted to mention, even though it's not a cichlid, the work by my student Bianca Brown, who has generated the first genetic data from a species of spiny blind eel in the lower Congo River. So you can see that these little guys are pretty helpless looking. They're completely blind, thought to be completely blind, pretty small. And there are a couple of different species that live in the Congo. Recently, Dr. Stiassny identified another species. About 3 years ago, she discovered another species. And we were curious about when that species had diverged from its sister species. So Bianca generated, again, the first genetic data ever for this species in our lab here. And using that, we estimated a divergence date for its nearest sister species of about 1.6 to 1.8 million years ago. And the Congo Basin is thought to be about 5 million years old. So this suggests, so these estimates are very similar for other estimates that have been done for cichlids. And it suggests that, first of all, that speciation is occurring in situ. It's not that these species formed someplace else and are washing into the lower Congo. They occurred in situ. It also suggests that all of these new ecological niches were opened up when the modern conditions that we see in the lower Congo were first formed. All right, so to bring this all together, these data, these are sort of the first wave of genetic data that we have for all these interesting new species in the Congo. And what they suggest to us is that, yes, this hypothesis that rapids can act as a biogeographic barrier is supported. Water can act as a barrier to fish if it's flowing fast enough and deep enough. Our next steps, as I mentioned, will be to move beyond simply looking for genetic differences across the genome and hone in on particular parts of the genome that may be involved in natural and sexual selection in these systems. But the overall message I want to impart to you is that the lower Congo River is this fantastic example of how a unique ecological environment can shape biodiversity, especially in a tropical ecosystem, and it's certainly one worth conserving. So I'd like to wrap up just thanking my collaborators, Melanie Stiassny and Jeff Markert, as well as some other folks that have been instrumental in collecting specimens and analyzing them in the lab. I'd like to thank PSD CUNY in particular for a little seed grant that allowed me to do this genetic work. And I'd like to thank you for your attention. I'd be happy to take questions.
Is it, I guess, based on your experience, would you say that the preference for, let's say, a color or certain fin type, is that more behavioral, psychological, or is that just a change in DNA and now all of a sudden they're attracted to a different color?
So that's a great question. In fishes, which have been pretty well studied in this regard, it often corresponds to a mutation that influences, for instance, a particular wavelength of light that can be seen better because of that mutation. So a mutation in certain genes that influence how a particular wavelength is perceived can all of a sudden open up kind of a whole new world for a fish, where they're able to see essentially colors that they weren't able to see before. great question. Another one? Yeah.
When you got, has anyone ever, or you guys, I guess, contemplate what would happen if you, if these species are very similar and they're genetically different, but like how long would it take for, like, if you actually, like, were to take some fish from one side and put them on the other side, if the environment, I guess, is suitable, can they mate it, and how long does it take for them to?
Right. How long does it? Right. How long would it take for them to become different enough that if you did put them back together, they wouldn't want anything to do with each other? Yeah. So that is sort of like the holy grail. That's the question that we want to answer for not just this system, but for all kinds of species. And the answer is it varies tremendously depending on the system that you're talking about. So in a the Crater Lake cichlids that have been so well studied, we see that kind of evolution occurring, that kind of speciation occurring over the course of less than 1 million years. So in terms of evolutionary time and geological time, that's like blink of an eye. Other species may take quite a lot longer to diverge. The general notion is that when you have strong selective forces, that's going to cause divergence to occur much faster. So if you have a species that's in a new environment that's quickly changing in alignment with that environment, then it will diverge, it will become different much, much faster than if you just have two species that are randomly kind of marching apart. Good question. Other questions? Sorry.
You find you have this rapid that is separated. What happens downstream?
So Bianca actually looked a little bit at that in her work on the Mastacymbalus, whether you get strong differentiation between upstream and downstream populations in addition to the cross-channel. As you would expect, you tend to see that fish can move downstream but not upstream, especially when you have these massive waterfalls that are blocking the way at certain points. So we can see species clusters at sort of different sections of the river and little mini species radiations in particular clusters that are separated by these waterfalls. However, in the eels, we didn't see a whole lot of upstream-downstream differentiation, actually, which suggests that they're kind of getting indiscriminately washed. So it may be that they're flowing downstream, and that's creating just gene flow all the way down. Yeah. Yes?
Can you say something about the mechanics of how you made those diagrams that show the differences between these species?
The haplotype networks. Yeah, so a haplotype network is just a sort of another way of drawing a phylogenetic tree, but it contains basically the same information. So you're taking different genetic types and arranging them into a cladogram that shows their relationships with each other. In the case of those networks, it gives you a bit of extra information beyond what a phylogenetic tree would because, I'm sorry, I think I didn't mention it, but the size of those circles is relative to the number of sequences that have that particular haplotype or genetic type. Does that answer your question? Yes? You mean from a social standpoint? Why would we want more species, or why?
I'd be curious, even from a biological perspective, why wouldn't that encourage increased speciation?
Well, so biodiversity we think of as a good thing in general because of, many reasons. Some are cultural, some are religious, and some are practical. The more species we have, the more ecosystem services we as humans are provided. For instance, there are certain species of bacteria that help us clean up oil spills, things like that. And we all know examples of particular aspects of biodiversity that help us as humans. So in that sense, we think of biodiversity as a positive thing for society and for our communities. But I don't know that anybody would think of the process of speciation as inherently good or bad. I haven't heard people talk about that so much. Yeah.
I'm interested in a different kind of evolution, that is your own intellectual evolution. I'd be curious to hear you talk a little bit about how you came to be involved in this kind of research. What made you be interested in biology and this kind of genetics? Talk about your own intellectual evolution.
Sure. Yeah. I've been sort of a closet naturalist since I was a little kid. But neither of my parents are scientists. My dad is also, he was sort of an amateur naturalist, very interested in, we lived in the desert and when I was growing up, and he was interested in scorpions and would take us camping and show us all kinds of things that made my mom nervous. I was lucky enough to have a few really wonderful teachers through the course of my career in junior high school and college that really encouraged my love of natural history. So I guess that's the main answer. It was, my interest in the natural world and the opportunity to get outside and explore all the interesting creatures that live around us that drove me to a career in biology. And I find evolutionary biology, no offense to my other colleagues in biology, to be absolutely the most fascinating subfield of biology. And to me, as I said at the beginning, the ideas underlying evolutionary biology unite all the other subfields. And I like that it's got some philosophical components as well. So there's a lot of highly intelligent evolutionary biologists out there who like to fight with each other, and that makes life interesting.
Actually participated in the collection in the rapids?
Yes. So, what's that?
How is that done?
So most of the collection is done from shore, as you would imagine, but there are some, in order to get some of the deeper water species, kayaker, or to get access to places where there's no access from the shore at all, either because there's no roads or it's just too dangerous. Kayakers, professional kayakers were part of the team to actually collect the fish. Yeah, with nets, yeah. And tragically, one was killed last year, not during the project, but in a subsequent expedition that was on the White Nile, not the Congo. It was killed by a crocodile. So it's a dangerous line of work. Yes?
It's very interesting talk and to see the influence of biology in the evolution.
Yes.
Do you only want to look at the chemical parameters in the water that we are finding?
That's a great question. I don't believe so. I know there's a field station in around Pool Malebo, where they may have done some work on nitrogen cycling and the like, related to annual flooding. But no, as far as I know, there hasn't been any in-depth studies of the water column. So I'd be happy to talk with you more about that. Yeah. Yes.
Has climate change been affecting these processes in the Congo at all? Do you know?
Right. Yeah. So that's a great question. Climate change in this area is thought to primarily be causing changes to the annual cycle of rainfall, and that certainly can have huge downstream, no pun intended, downstream effects on everything that lives there. So this cycle of flooding that we were just discussing has an important role in particular for juvenile fish, which tend to be born and live at sort of the edges of flooded area in sort of a safer zone before they become big enough to inhabit the main channel. So changes to either the amount or the timing of that flooding could potentially have a big effect. All right, thank you very much for coming.